Welcome again to the Gable Hall Science Department channel. I will be talking about food tests today. We'll run through the test for starch, the test for sugars, the test for protein, and the test for lipids. First of all, let's start with the test for starch. If you want to test for starch, for example, I've taken one of these biscuits and it's in the mortar, and I'm going to hit it with a pestle to break it into little bits. Then I can take a bit of my biscuit and place it on the test well. Okay, to test for starch, I'm just going to add iodine to the biscuit. If it goes black, like you can see, it means starch is present in the biscuit. So that is a test for starch. It's quite simple. It's a straightforward test. You add iodine to it. If starch is present, it's going to go black. Now let's go on to the second test. That is the test for sugars. Okay, specifically the test for glucose. For the test of glucose, you use the Benedict's solution or Benedict's reagent to test for glucose. And you crush your sample as usual. If your sample is a liquid, you don't need to crush it, obviously. But if it's a solid, you crush it. And then you place a bit of your sample in a boiling tube. Okay, shake and then add some Benedict solution to your sample using a pipette. Now the solution is blue because the reagent is blue. If you place the mixture of your sample and your Benedict solution in hot water, boiling water for a while, let's say for about 5 minutes, if sugar is present, after 5 minutes, the solution is going to go, if there's a little bit of sugar, it's going to go yellow or orange. If there's a lot of sugar, it's going to go brick red. If there's still a little bit of sugar, it might go green as well. So if there's just a little bit of sugar, it's going to go green, orange, or yellow. If there's a lot of sugar, it's going to turn brick red. So just let's leave that there for 5 minutes and then we can look at it later. The next test is a test for proteins. And the protein test, you use a biuret solution. With the biuret solution, I will do the same thing. Crush my sample. As usual, if the sample is a liquid, you don't need to crush it. So I've crushed my biscuit sample and I place it in my boiling tube and I add biuret solution to it. Now, if I shake the sample for a while, let's say about 2 or 3 minutes, if the color goes pink or purple, then it means proteins are present. So if it goes pink or purple, it means protein are present. Just to recap on the protein test, you crush your sample again, you mix it in your boiling tube, you add biuret solution to it. If it goes pink or purple, then protein is present. We've talked about the test for starch, we've talked about the test for glucose, we've talked about the test for proteins. Let's go to the last test, which is the test for lipids. Now the test for lipids, if you take your sample and place it again in a test tube this time. So I get a bit of my sample and I place it in a test tube or a boiling tube. If it's a liquid as usual, you don't need to crush it. And then you add some Sudan 3. So you use Sudan 3 solution for this test. And you add your Sudan 3 to that sample. And then you shake it to mix together. If lipids, which are fats and oils, are present in your sample, then there will be a distinct red layer which is formed at the top. So a distinct red layer will be formed at the top of your sample. That tells you that lipids are present in that sample. I hope that helps you very much. That is a test for starch, for glucose, for proteins, and for sugars. Don't forget to click, to share, to subscribe in our channel. Thank you for listening.